Okay, I'd like to welcome everybody to our first seminar of the 2025-2026 series. I really appreciate everybody coming today. It's great to see such a good turnout. We have a talk from France on cancer immunology today. It should be exciting. As always, I need to remind people that these seminars are live streamed and recorded and available archivally on YouTube, so please be aware of that. As always, Tom Helikar, myself, Jim Sluka, and Lorenzo are available for suggestions, critiques, comments. Please feel free to reach out to us to tell us what we can do better. We have a variety of communication channels. They work better if you use them. We tried to create some QR codes for them and make things a little bit easier to reach. Feel free to help us publicize the seminar series. We'd appreciate it. We're trying to integrate this seminar series with other groups. In particular, we have our friends who are doing RDA, digital twins. We have our friends in Canada and others trying to work together in the area of computational modeling applied to biomedical problems. If you have a publication to share, we can put it up on the IMAG site and also on the GLIMPRINT site. Please send those to Jim Sluka. We'll do our best to get you the attention that you need. The GLIMPRINT site also lets you list upcoming events, job postings, funding opportunities, anything that you think would be of interest to the community. Please let us know. Coming up in the US, October 20th to 23rd, University of Wisconsin-Madison, there is the Combine meeting. That's of interest to a lot of people in terms of computational infrastructure to enable large-scale models. It's officially still allows submission till September 28th. One of the great things about Combine is it's a little bit informal, so you have a lot of opportunity for late scheduling. Please do think about that. The upcoming one is in Auckland. It's a little further for many people, certainly easier for some people, but people should be aware of that. And I know many people in this community participate in that. We are going to have a virtual celebration conference on October 16th and 17th to celebrate the 5th anniversary of this series. We have as confirmed speakers Gary Ann, Jane Bai, Francois Grenier, Tom Helikar, Nicole Kleinstroyer from NIH, Reinhard Laubenbacher, Grace Peng from NIH, and Reid Schabman also from NIH, Sarah Teichmann, Tom Yankeloff, and others. You'll recognize many of these names as former speakers. We're still working to line up additional speakers for this two-day meeting. We'd very much appreciate your attendance and help publicizing this event. There are going to be opportunities for panels and discussions as well as presentations. We realize it's a little bit late to be pulling it together, but we want to make sure that it serves the community and people know about it. Coming up, we have seminars scheduled for the 18th and October 2nd. We would appreciate suggestions for the October 9th date, and then we have that big meeting on the 16th and 17th. As always, please suggest speakers, suggest yourself, suggest your students or postdocs, suggest the people that you would like to hear from. We depend on you to get us lists of speakers that you want to hear so that it's productive for you. And don't be shy if you've spoken already and you're doing new work, you want to update us on where things are, you want to tell us about another project, we're happy to hear about that. And again, we have people of all levels of seniority here. We've had many talks by graduate students and many talks by senior professors. And so don't feel shy about suggesting people at all stages of career as possible speakers. We're also happy to have suggestions on experimentalists, on industrial or biotech people who might speak, anything in this broad area of multi-scale modeling, immunology, digital twins. We can think broadly, so please do suggest things for us. We really depend on that. Tomorrow at 11 a.m., we're going to have a steering committee meeting. That will be to finalize the schedule and publicity for the virtual workshop on October 16th and 17th. We would very much appreciate your help if you want to come. If you think you should be invited to the steering committee and you haven't received an invitation yet, please let us know. We're always happy to have people join us. Are there any other announcements before we move to the seminar proper? Okay, today we are hearing from Malvina Marku from Toulouse University Cancer Institute, who's going to be talking to us about multi-scale computational modeling of tumor-immune interactions, which is certainly a topic of great interest to this series. So I'll turn the screen share over to you, and I want to thank you again for being willing to speak to us today.
Hello again, everyone. So I'm very happy to be in this series of seminars and to present some of the most recent work that we have been conducting in the Cancer Research Center of Toulouse, focusing on applying some data-driven approaches to infer or to build dynamical models in the molecular scale. So perhaps in this effort of building multi-scale dynamical models, combining different levels of regulation, I'm going to go a little bit of a step back, focusing on the molecular scale and investigating them, some approaches, some data-driven approaches to build dynamical models in this scale. So in the molecular scale, what we want to understand in general, it is the investigation of mechanism cellular phenotype, and like what causes a cell to become another cell, or to change its phenotype, or to behave in a certain way, and so on. So imagine a cell, for instance, being in a given environment. Then there are many multiple ways that the cell can interact with the extracellular environments, through the exchange of different signals, through the activation of different ligand receptor pairs, and so on. And then this signal is then transmitted to the gene level. The regulatory interactions between these genes can give us then a different phenotype of the cell. This can be imagined like a cell, for instance, passing from state 1, that it was initially, for instance, then the receiving the information going to state 2, and so on until it reaches a phenotype. And this is the core, I think, of the gene regulatory network, dynamical modeling of gene regulatory networks. It's a very simple and simplified representation of what is happening in reality. But in general, just for the sake of like giving some very general information, but I think that the audience here is very familiar with, in this representation, in this network representation, the nodes of the network are the genes, and in very general terms that can interact with each other in two ways, either activation or inhibition. And then we can apply dynamical models like to see how these interactions can change in time and then leads to different phenotypes. But the question is, the question that I pose it is what do we do for building this, the gene regulatory networks and the dynamical models that explain this network, that study these networks. Well, what we do in general, it is that we gather as much information as we can from different types of sources, like from literature, from public databases, experimental data, or even data from patients when they are available. These sources can be generally categorized into main groups, like the prior knowledge sources or the data-driven ones. But whatever the source or the combination of the sources, what we tend to do, it is to build the general model. And then when a patient can come with its own profile, with its own molecular data, then we can personalize the model and then use this model for different purposes like prognosis or therapy optimization. But this general model, well, it has the merit of capturing different the heterogeneity in different contexts, in different data, and different patients, and it should be general enough to capture this heterogeneity. But then combining different sources can help us have a more comprehensive understanding of the system that we are studying, and it has the facility of dealing with incomplete data when using only one or some of the sources. So yeah, there are some positive aspects of the general model, but there are also some challenges with it. Like in this very simple representation in the gene regulatory network, we try to squeeze in different levels of control. We don't capture here first, or we don't represent here the chromatin state or the 3D organization of the chromatin or the transcription factors, how they interact with each other, the signaling path, the level, and so on. So it is very dense representation, very simple representation of a very dense information that we should gather. And the other aspect, from my perspective, the most important one, it is that these models are very context specific. Means that if we want to build a GRN and a dynamical model of a cell, but in different conditions, in different tissues, and in different individuals, these models will be different. And let alone here, the different cell types that, of course, the models are going to be different. So having a general model lacks a little bit or misses a little bit this context specificity. And another very critical point from my perspective is the fact that from using prior knowledge, like public databases and literature, we tend to limit ourselves in studying some very known genes, interactions, or pathways. And we try to focus only on these interactions that are very well studied, but we can miss information from the shadows, like what is not so well studied. So there are some drawbacks as well from building these general models and not following a data-driven approach. So the question that I pose is, are we looking under the lamppost when we build the model, like focusing again on these very well-studied pathways, interactions, and molecules like genes and transcription factors? And some of the questions that I want to discuss in this seminar, it is actually the challenge that we took in our lab to take the data-driven approach, the challenge of following a data-driven approach and using longitudinal data, like molecular longitudinal data, like expression data, to build context-specific models. And the question that we can discuss is that how to combine actually the data-driven with the prior knowledge, like in order to get a more comprehensive and a more complete understanding of the system that we're going to study. So in Palcaldi's lab at the CRCT, Cancer Research Center of Toulouse, where I was conducting my work, we studied the tumor microenvironment. Very brief concept, like definition of it, but you are all very familiar with. To my perspective, or to my understanding, it is a ecological system composed of different cells, of course, cancer cells, but a lot of other immune cells like macrophages, NK cells, T cells, dendritic cells, and blood vessels as well. And it is well studied now, very well documented, the fact that the interactions between cancer cells and immune cells can define how the system is going to go, like whether the cancer is going to grow or not. And in Palcaldi's lab, one of the cell types that we were particularly interested in are macrophages. Like most of the other immune cell types, these are very plastic immune cells. They can go from pro-inflammatory to anti-inflammatory. But the particular characteristic of the cells is that the macrophages that are found in tumor, which are called tumor-associated macrophages, can have pro-tumoral roles that can help the cancer cell from going to apoptosis, but they can also trigger the function of other immune cell types. So it was a particular interest to study the phenotypic shifts of macrophages and how this phenotypic shift can lead to tumor resistance. And the tumor that we were interested or studying mostly, it is chronic lymphocytic leukemia. Briefly, it is a blood malignancy characterized by malignant B cells that proliferate into the lymph nodes, but that they circulate in the blood. So there are a lot of interactions happening between these CLL cells and immune cells in both compartments. And I was explaining a little bit the fact that the interaction between cancer cells, CLL cells and immune cells, it is now very well documented. There are different ways, ligand receptor activated and so on. So the project at our lab was to build these multi-scale models of immune cell and cancer cell interactions, combining the molecular scale with the cellular scale. So some past work that we have done in our lab, it is first to study the polarization, the phenotypic shifts of the macrophages. And for this, we built a gene regulatory network of macrophage polarization, this being in different contexts like pro-inflammatory signals, anti-inflammatory signals, or the signals coming from the CLL cells, like the cancer cells. This was a literature-driven model built actually from some previous work published, and we extended it to include also some CLL signaling. And we were able to identify distinct phenotypes of macrophages, the M1, the M2, and the pro-tumoral phenotype. And we used then the model to identify which molecular nodes or molecular targets should we target in order to stop the formation of these pro-tumoral phenotypes of macrophages. And in some other previous work, we also built another model in the cellular scale, like to model the interaction between the macrophages and cancer cells, but this time in the cellular level. So we built an agent-based model of macrophage cancer cell interaction. Here in this diagram that you see in the central figure, there are different changes in the phenotypes of both immune cells and cancer cells and the way that they interact with each other. And we used some in vitro cell counts measures that we produced in our lab to optimize the parameters of our system and thus of our model, and thus identifying which are the biological processes that were the most determinant for the profiles that we were observing in the cell counts measures. So we identified actually that the initial state of the cancer cells, but as well the phagocytosis performed by the macrophages toward cancer cells, as well as the anti-apoptotic signals secreted from pro-tumoral phenotype of macrophages were the most determinant for what we were observing in cell counts. So the challenge that we had to complete the model was also to build a dynamical model in the molecular scale for the cancer cells, but this time following a data-driven approach. So before going to the results, I'm going just to speak a little bit about some paradigms for network inference methods. So there are different approaches or different paradigms, but they can be categorized in two main, I would say, groups. First, it is the expression-based network inference methods, in which the expression data, it is the primary source of information. And then auxiliary data can then be used to inform which is the structure of the regulatory network that is going to be inferred, that is going to be modeled. And in the second category, the predictive model of expression, actually the information it is taken from usually from enhancer promoter level, in order to explain the expression data. So the expression data is used as an output, actually, like what is the model explaining or trying to explain. And in this, the tools or the approach that we took in our lab was in the second, or like falls in the first paradigm, like expression-based network inference methods, like using expression data to infer who regulates what, basically. So in this paradigm, as I said, like the input consists of the gene expression data, RNA-seq or microarray data, and then different algorithms can be applied for inferring, again, the structure of the network. This can be molecular networks, this can be mutual information methods or differential equation or probabilistic models. And the output, it is going to be a directed network indicating, again, or trying to identify the covariates explaining the expression of a target gene, like what regulates what, who regulates what, basically. And as I mentioned, that auxiliary data can then be used for helping or for improving the inference of the structure of the model. This can be, for instance, different type of input data, for instance, attack-seq or sequence motifs. And then some additional analysis that we can perform on the input data, for instance, the estimation of the TF activity, again, with the main scope, but trying to improve the inference process. So a very critical point, like while quantifying the performance of a certain method can be very standard, like estimating the standard measures, like for instance, area under precision recall curve and so on. A critical point in the inference of the gene regulatory networks following a data-driven approach, it is its validation. Like how do we validate that the network that we have inferred, it is an accurate one, it is what it is actually happening in the system. So different ways can be applied, but I mean, all of them has their pros and cons. For instance, like using public databases, We can validate the interactions using these databases, but we're going to be limited to highly studied pathways. For instance, if we have inferred interactions between two genes, but this it is not so well studied, so it doesn't exist in public databases, that is going to be considered as a false positive from our method. We can compare to random networks, like how our network performed to compare to random ones in public databases. And the most complete one would be, of course, the experimental validation. Like, for instance, in this example, we have inferred, let's say, that the GLI3 here, the central node, has a downstream effect like it regulates the expression of this downstream TFs or genes. And then we can go to the lab and then we perform some perturbations of this central node and see that indeed there is some changes in the expression of its downstream targets. But then, of course, we can imagine that with all the combinations of all the genes that we should perturb in order to validate the network, that can be easily extremely expensive. In general, all the inference methods suffer from a high percentage of false positive interactions, and this issue has been discussed in in different papers published. Like different methods trying to cover as much as possible interactions between genes tends to have like this, in this way, a very high percentage of false positive interactions. But despite all these challenges, the data-driven inference as a discipline has been an ongoing field now for more than 25 years, starting from Bayesian networks, reaching a milestone when some other algorithms were applied to infer actually not only one network or one model, but an ensemble of models. And then in 2016, starting with using of single cell for inferring the GRNs, and then combining this data like single cell RNA-seq with ATAC-seq data, and nowadays using all types of advanced algorithms like transfer knowledge, graph neural network, evolutionary neural networks, and so on. And we try to summarize some of these methods in the review that we published 2 years ago. So going back to our problem, or to the project that I was working on, again, the scope was to study the interaction between immune cells and cancer cells and trying to build a molecular model of the cancer cells following a data-driven approach. So for this scope, we went to the lab and produced our own data. So we had a extremely limited number of patients that we were having, like taking the samples from. Well, we had only 3 patients, but we performed 2 different conditions, in vitro conditions, trying to, again, like to study the interaction or the role of the presence of the immune cells in the viability of cancer cells. We performed 2 different conditions. In the first one, culturing cancer cells and immune cells together. And in a second condition, which is the monoculture, we separated the cancer cells and let them in the culture alone. So we could see how much the immune cells can play a role in the survival of cancer cells. Both conditions, cultures were led for 14 days, and 5 time points were extracted. We extracted the RNA from 5 time points and performed bulk RNA-seq. So we have a longitudinal RNA-seq in 2 different conditions. So there were some expectations or some hypotheses that we were drawn. What we were supposed to observe actually in the dish, like first we would see some adaptation in the first phase, like isolation stress. This also was seen in the expression level. And then in the autologous culture, we would expect that, of course, there are a lot of immune cells, so there's going to be a lot of interactions happening between cancer cells and immune cells, and within immune cells as well, and of course some other phenotypic shifts happening within immune cells. And then as a final state, if the environment supports the activation or supports the proliferation of cancer cells, it's pro-tumoral, then the cancer cells were going to proliferate or at least activate some pathways of proliferation. Otherwise, they're going to go to apoptosis. And we wanted to study the cell fate of cancer cells through the network representation. Again, having different signals coming from the extracellular environment, we wanted to have or to infer this gene regulatory network, then can be used for describing or for capturing the different cell fates of cancer cells. So there were a lot of bioinformatics analysis that we did prior to the inference of the gene regulatory network. I call this phase as learning from the data. First, we looked just at the viability, which is the percentage of alive cancer cells remaining in the dish in the two conditions. In goldish, you see the monoculture where the cancer cells are alone, and in bluish, greenish, it is the autologous culture. So we see that there is a decrease actually in the viability of cancer cells in the monoculture in the latest time point. But you see also that there is a lot of heterogeneity between patients, like not all the patients respond equally. And this is also seen in the transcriptomic level, like just doing a PCA with all the patients and the two conditions, we see a clear separation between patients. But within patients, we see also like a difference in like separating the autologous from the monoculture. And this was also seen in the pathway level. We performed gene set variation analysis. We see consistently that in the three patients, there are a lot of pathways. If you could read, there are a lot of pathways related to the response to immune system, response to immunity in the three patients that are more activated in the autologous versus monoculture. But again, I mentioned the fact that the three patients are very heterogeneous, and there is that not all of them respond equally. So then another analysis that we performed prior to the network inference, it is to perform some ligand receptor analysis. So in this approach, the expression data that we have, it is used as the fingerprint, like what it is, the downstream of the activation of the ligand and receptor. So we have the ligand receptor first, and then what we see in the expression level, it is actually the result of the activation of this ligand receptor pairs. So we performed ligand receptor analysis in both autologous and monoculture separately. We see surely that in autologous there are way more ligand receptor pairs activated, and particularly the two ones that I'm highlighting here are two very important ligand receptor. These are ligands that are involved in the interaction between cancer cell and pro-tumoral macrophages, which were present in the dish. And this is also seen in the pathway level. Again, you see, for instance, interleukin-10 signaling that it consistently more activated in the autologous versus monoculture. And then going to the inference of the gene regulatory network, again, what we had, it is a time series of RNA-seq. And as a first step, what we did actually, it is to reduce as much as possible the complexity of the network that we're going to infer by focusing on the TF activities. Transcriptional factors. And in transcriptional factors, as the expression is not so much representative of them, we estimated the TF activity using the collect tree network, and Viper is a tool that we can use for estimating the TF activity, and selected from there the TF, the transcription factor that will change in their activity the most during the time series, like either going up in the activity or going down in the activity. This performed separately for each of the patients and each of the conditions. So we had a group of the transcription factors that were identified as most differentially activated, this being up again or down, and performing some pathway enrichment of these TFs and their targets. We could see a lot more activation of these pathways in the autologous culture compared to monoculture. Here, for instance, in the upregulated TFs. These are pathways, actually very important pathways involved in the response of the cancer cells toward immune cells. And then again, the main scope, it is to build the gene regulatory network. So for this, we combined two different approaches, or two different ways. First, it is that we used DIN-GNE3 method. It is only one of the many available tools of to infer gene regulatory network from using time series of RNA-seq. This is a dynamical version of GNE3. GNE3, perhaps might sound familiar to you. It is widely used also in single-cell GRN inference. Basically, in this method, the expression of a certain gene at a certain time point, like Xi, for instance, at time t+1, it is modeled as an ODE function of its regulators minus a certain degradation rate. And then the tool implies a random forest algorithm to infer or to learn these functions. And then it will associate a weight of interaction, estimated from the variance of the learning sample. So in the end, what we're going to have, it is it is going to be a directed and a weighted gene regulatory network. And then we combine this this approach with just the TF activity correlation, like having again the activity of the TFs. We just estimated the correlation between them. Thus, also having the information about the sign, assigning a sign of interaction, like whether if the correlation was positive, that we were assigning a positive sign, if not, then a negative sign of interaction. So with this, all I'd say like the backbone of the analysis that we did, the final gene regulatory network looks like this. So again, it's going to be a directed, signed, and weighted GRN. Just represented like this, it doesn't say so much, it's just like a hairball, but then we started to map some of the information that we have from the context, from the culture, the two conditions that you have, different patients and different time points that we were extracting these TFs. So we performed some network analysis. Particularly, we estimated the associativity of different features. Associativity being a measure of how much, what is the likelihood of a node having a certain feature. For instance, here being either the condition or the patient or the time point where from which the TF it is extracted from. What is the likelihood of this node to interact with the nodes with similar or with the same feature? So the associativity is going to be a value between -1 to 1. A positive value indicating that the nodes with similar feature or with same feature tend to interact more together than with other nodes having different features. So for instance, from mapping the condition in the network, here you see, for instance, again, in goldish, it is the monoculture, and in bluish, it is the autologous culture. We could see that the network has a high associativity of 0.4 compared to random networks. And it is more or less the same thing with other features like the patient and the time annotation, the time where the TF, from which the TF it is extracted from. Like this saying that, or noting the fact that the network that we have inferred has a very modular structure. And we went a little bit further actually with to detect the modules, the main modules of the system. We performed this by using cluster Vs. It is a plugin in Cytoscape. We detected in total 11 different modules, with the two biggest ones, the first one here and the first one here. Actually, by performing some pathway enrichment of these TFs and their targets, we could see that the first biggest one, it is a module related to like some basic processes happening both in monoculture and in autologous culture, like for instance, regulation of cell cycle, regulation or response to DNA damage or cell fate decision. Whereas the second one, it is mostly related to response to the immune system, like for instance, regulation of cytokine, response to the immune system. This performed again, highlighted by the by performing pathway enrichment of these TFs and their targets. And the most peripheral modules then are very patient-specific and time point-specific and correspond to very particular biological processes happening at particular time point and a particular patient. So this is actually where our work finished, but of course it is not done yet toward the final goal, which is to build a dynamical model. So this work is still ongoing in Pancaldi's lab. We started exploring Colomodo, which is the consortium of logical modeling tools. Over there are a lot of tools that we can use for dynamical modeling, particularly Boolean modeling or logical modeling of of gene regulatory networks. But there are also some tools over there that can be used for inferring the dynamical models given expression data or just given the structure of the network, the topology of the network. For instance, Bonesis, which was one of the tools developed from one of our collaborators, what takes as an input is the regulatory network, like the gene regulatory network, and some constraints that we can put as an observation. For instance, the binary state of the TFs or the nodes in the network at different time points, the existence or the absence of a given trajectory, the existence of absence of a certain trap space or a fixed point, and so on. And then it is going to learn, it's going to infer all the possible Boolean functions or Boolean models that explain these constraints or observations that we put as an input. And then again, just saying that the final goal will be to integrate this Boolean model of cancer cells to an agent-based model, like here combining the molecular and the cellular scale. This with to then be used for the common scopes, like to identify the drug targets, to perform perturbations, and so on. So with this, some perspectives and some future work that, in fact, this is the work that I have just started. It is the fact that when we build these dynamical models or Boolean models in general for the molecular scale, we can use them for performing perturbations or performing overexpression or knockout simulations. And it is easy in this way to identify which are the nodes to be perturbed in order to have a desired effect, like, for instance, apoptotic cancer cells or abscess or proliferation and so on. But what we tend to avoid in this identification of drug targets is the fact that the drugs actually that target these genes can have a wide range of side effects, from mild to adverse side effects. They can be very toxic. So my scope or my work, the work that I have just started now, it is to combine actually or to optimize the search of novel drug targets, taking into the consideration the toxicity that a drug or perturbation can bring to the system. And I am happy that I have just started actually this new postdoc position with my new mentors, Etienne Chatelud and Melanie White-Koning in the Cancer University Institute of Toulouse, with the big aim of building digital pharmacological twins. Again, combining different scales of regulatory scales in the modeling part, molecular and cellular, and this to be combined with pharmacokinetics and pharmacodynamic modeling. So I conclude here with some conclusions that actually we discussed I discussed during the slides, and most of the work is done in Pancaldi's lab at the CRCT, but there are also a lot of help that we got from the consortium that we had with Loïc Polevet in Bordeaux and Laurence Calson in Institut Curie in Paris, and Andrei Zinoviev at Evotech here in Toulouse. They were funding also my work. And I thank you for your attention, and I am very happy to take questions. I see Lorenzo has raised his hand.
So Lorenzo, go ahead, and then
perhaps I might stop sharing.
Oh no, that was fine. All right, thank you, Malvina. It was very nice. Especially I like the idea you started with expression data. You chisel down with a number of tools to get to that map you have toward the end. My question is exactly on that map. I noticed that there are, at least by peeking at it, there are a few nodes that seems to be kind of funneling some other stuff, but they are eventually like hubs. Have you tried to isolate those and try to see if you can simplify further the map and see, all right, eventually the signal goes around these, I don't know, 20 interactors that are funneling the signal over the other, I don't know, 100 that you have there.
Indeed. So in fact, there are some, I didn't show here, but there are also some other measures that we measured, that we computed for the network, like between a centrality of the nodes and other centrality measures. So we could identify actually that there were some nodes with a high between a centrality or with a high centrality in general, like not only between us. These were actually the nodes that were in common between different conditions and within different patients. Like they were serving as links with very isolated modules afterwards that were explaining a very particular or specific process happening to one patient at a particular time point. So we didn't perform any reduction from the network. that we inferred, because we wanted to preserve like what is the information that these TFs, these central TFs, were trying to link. Like it is this very particular or specific biological process happening in the periphery of the network, linked by a central node, and then perhaps it is transmitted to the core of the network or vice versa. We wanted to preserve actually this information coming from both ways, and we did not reduce it further.
Okay, thanks. Thank you very much.
Heiko, go ahead, please.
Yeah, hi. Very interesting talk. Thank you so much. It looks like a lot of your analysis was done on 3 patients. So how robust are these networks and directions you're getting out of your analysis, given this high heterogeneity in gene expression that you have shown? If you get additional patients, will this completely change, or can you comment on that, please?
Yeah. It's thank you for your question. It is a question that perhaps I might not have the answer. Like I would say, like if I were to pose the question to myself whether this is a general model of CLL, I would say that it is not a general model of the CLL. So if a new patient comes, for instance, that I can personalize it to this new patient, because this modular structure that we observe from different patients at different time points might indicate that different regions of the network actually are very patient specific. So this might be the cause of the very limited number of patients that we have. So I cannot generalize it to other patients that might come. Of course, they might depend on their profile as well. So perhaps just looking at the expression, like how similar the patient can be, the new patient can fall to patient 1 or patient 2, patient 3, but it can be also very different from here. So to be fair, I would not take the big step to say that, yes, I can personalize it if a new patient comes with their own profile.
So if every patient comes in with their own profile, and is this methodology then really identifiable, or is it of high risk of overfitting or detecting noise in the data?
But again, like I would say, like it depends on the profile of the patient. So I cannot give an answer for that, to be fair, because it all depends on how the patient look like, and they're very, very different from each other again. So I don't know. Sincerely, I don't have the answer for that.
Yeah, no, I mean, your answer is exactly the concern I had. Thank you very much.
Thank you.
Is there a question from Naam?
Yes, this is me. Thank you for your talk. Very interesting. The question I have is related to the model you use itself. I mean, CLL is actually not a solid tumor, but you are actually doing your modulation or your modeling with macrophages. Macrophages are typically for solid tumors, and I wonder how your model would actually perform if you are actually using a solid tumor in which you do have this type of interaction between macrophages and the tumor itself. I know that in CLL, for instance, is a liquid tumor, is a leukemia, and it's very predominant actually. There is some type of interaction between macrophages, but most of the time it's actually the differentiation of monocytes into macrophages that actually rule this kind of interaction. What happens if you actually use this model that you are using into a typical solid tumor, saying whatever the tumor you want to choose, and see how that actually performs? Thank you.
Thank you. Thank you for your question. So, in fact, the system, in vitro system that we have, it is true that it is a liquid tumor, chronic lymphocytic leukemia. So we have blood samples from patients. It is liquid. But actually, the system, at least what we saw, what we are observing, it is the fact that the system itself has a kind of mixed profile of liquid and solid, because it doesn't flow, so it is static. We know, or there are many publications indicating the fact that the CLL cells do not proliferate in vitro, but we did see some activation of proliferation. So this is very typical of lymph node. It is not of blood, actually. So we were thinking that perhaps the fact that the system, it is static, might be a little bit more similar to the lymph node than to the blood where it circulates. The formation of monocytes and macrophages and the interactions as well between them, it is more typical of the solid tumor than to the blood. We don't have macrophages in the blood. So it has a mixed profile, I would say, between the two. So we do not have samples from lymph nodes, but I'm curious, like actually, like to see like whether it's going to be more like a little bit closer to the lymph node than to the blood, actually, like interactions.
Hi, this is Yi Jiang from Georgia State University. So if I understand you correctly, you have for each patient, you derive one network, and that network is different between patients.
No, in fact, we generated or we extracted the transcription factors for each patient separately. But then the network, it is used by using all the data from the 3 patients all together. So it is not a network merged from different patients, but inferred from the 3 patients all together. And then the modular structure that we observed actually were that different modules were explaining, or some of the modules were explaining particular processes happening in different patients and in different time points.
Right. So my question would be, if you had generated network for each patient separately, how different they're going to be? And is there a way to systematically, more rigorously test the sensitivity of your model?
Exactly. So thank you very much for the question. We have tested it. But we were limited because the number of samples that we have for each patient, like to be statistically relevant for the inference itself, like in the inference methods, the more data that you have, the more samples that you have, then the more statistically significant or relevant is going to be the interactions that are going to be inferred. Whereas using each patient, at least this was our case, then using each patient separately would limit the number of samples available for the inference. So we tested it, like performing the inference for each of the patients separately, and then we merged them together. It did not have the same structure. But we were assuming that it was basically the limit number of samples that we have for each of the patients separately to perform the inference.
Then I'll follow up with a little bit. If your goal is a digital twin, then you want a patient-specific network. So there seems to be a contradictory.
It is true. It is true. I mean, like, I don't know. I mean, like when we build the models, again, the 2 ways that we can go is either to build a general model and then personalize it when a new patient comes in, or to build a model for the patient from scratch. So we have all the infrastructure, the computational infrastructure, and we build a model for these patients. What we tried to build, again, we were limited in the number of patients, but we would try to build as much as possible a general model for the 3 patients that we had. And with it was the question before that if a new patient comes in, then we can try to personalize it. I mean, perhaps it was not in our case because we were very limited in the number of patients, but my approach, it is to build a general model in the beginning and then personalize it with a new patient when a new patient comes in. At least this is my approach, but I am open to discussion for that.
I want to press on this a little bit more. If you believe each patient will have his or her own network, what does it mean to have a general network?
Again, it depends on how do you perform the personalization. Either it's going to be a separate network or it is going to be the same network, but the expression of the nodes that you have in the network is going to be different from one patient to the other. So this is the 2 different approaches that I was trying to explain. My approach is that I want to build a general model, and when a new patient comes in, it is going to have a different profile. Thus, the expression of the TFs or the expression of the gene in the network is going to be different, but the network, the core, the structure of it is going to be the same between patients. So this is going to make the difference between patients. But again, it is also the other approach that you have a network for a patient, a specific network for a given patient. So again, what we were trying to do, although we were limited in the number of patients, was the first one, like to build a general model and then personalize it with a new patient.
Thank you. Thank you.
Thank you.
Okay, other questions?
I'm just kind of curious about that last point. Sorry, the camera's not up right now, but thank you for the talk. It was quite interesting. I'm just kind of curious if you had all of these different patient-specific networks. I realize that you said that you're trying to have a single structure from which all these ones, these other personalized versions come from. But if you were generating all these different networks, are they like truly sort of independent networks that you're sort of unioning together, or are they all essentially sort of like subsets of I guess like a more full system? Because if you say, oh, well, I'm kind of like zeroing out this, perpendicular subset, I guess, for patient A, are they all coming from like a same sort of universal network, I guess I should ask, if that question makes any sense.
In fact, I am not sure that I understood the question.
Oh, okay. I'll formulate it better and maybe I'll email you afterwards. I'm just wondering if these different networks can be sort of unioned together and say like, oh, actually they're all kind of like coming from a more general network.
With different networks, to refer to patient-specific networks.
Yes.
But again, I don't have a patient-specific network. The network, it is inferred from the data from the 3 patients together. So of course, if I had 10 patients or 20 or 100 patients, that I would infer one network for all the patients that I have. This is, again, very limited in my case, but it would have been a general model explaining the data or explaining the patients that you are using as an input.
I see. Sorry for my confusion. Thank you.
No, it's okay. It's just yeah, I know that. I mean, like I'm mixing a little bit the fact that the network itself is very modular, so there are modules with very patient-specific profiles, but the network itself, it is the general model of the 3 patients that we have. So we tend to build this general model of containing all the patients available.
So the network model itself is general, but then it's personalized by the parameterization, and those may lead to very different functional architectures.
Yeah, so it's general, again, like with very, very precaution here to say that it is general because we have only 3 and the 3 patients are very different, have very different profiles. So whether it is general enough to be used when a new patient comes in, for instance, with a completely different profile, does it fit to this network or not? So this is a question that we still do not have the answer because we don't have more data than this, but it is. So it is general for this amount of data that we had, but I would be careful to say that it is general enough to be used widely.
I just meant that it was explanatory for the data sets that it was created for.
Yeah, exactly.
So it's comprehensive over the data sets you used to create it.
Exactly. Yeah.
Okay. Are there other questions?
Just a curiosity if it's easy to scale up. So let's say that someone data bank or a biobank give you 200, 300 patients that you can use for building a better network. Is it easy to implement in such a way that you can easily scale up?
Thank you for your question. I never thought about this actually because the system that we wanted to study, I mean, like the data that we had, it's very, very context specific. In terms of computational, let's say, workflow that we follow, it is not so computationally expensive, so it can be definitely adapted to wider data sets. Perhaps not all the filtering that we performed initially. I mean, like all the analysis, the bioinformatic analysis, perhaps it is not going to be relevant for a wider a wider scope. But the selection of the transcriptional factors, the more differentially activated ones, and the network inference itself, it's not computationally expensive, I would say, so it can be scalable computationally.
Okay, well, I want to thank you very much. I think we had a lively discussion, and I hope we'll have you back maybe in a year when you have more patient data, and we can see how these methods generalize and be able to answer some of the questions
Exactly.
that were raised.
Exactly,
Was-
or perhaps with new results with linking molecular scale with PKPD modeling as well, which I'm starting to work on. I'm very happy to be working on this field, and I hope that I'm going to have enough results for sharing with you soon.
I'm personally very interested always in integrating these kinds of models inside of our multicellular frameworks. Right. If we have things like for CompuCell, if it's written in MapBoss, we can run it inside of CompuCell simulation platforms like PhysiCell can as well. And so it's really exciting then to think about what you could do to integrate in that multicellular context that you showed earlier. So that's something else we could maybe talk about going forward. All right. I want to thank everybody for joining us today, and I will see you in 2 weeks.